Hello everyone, my name is Gary O'Mealy, and in this video we are going to discuss passive transport. First, let's talk about simple diffusion. Simple diffusion is the type of membrane transport that we introduced in the previous video. This is going to involve small, nonpolar molecules moving freely by squeezing in between the gaps in between neighboring phospholipids. In the previous video, we of course discussed some of the rules that dictate which kinds of molecules prefer to do this. So this, as we said, is going to involve molecules that can freely pass through the membrane because they are small, because they are nonpolar, because they are electrically neutral, and they're going to be moving back and forth from either side of the membrane at a given rate. The first discussion that we're going to want to have here is talking about the rate or speed of this diffusion across the membrane and how there are different specific factors that are going to affect what that rate is, and those factors are defined by Fick's law of diffusion. The first factor that is going to affect the speed by which molecules move across the membrane is temperature. So the idea here is that molecules are going to have greater amounts of kinetic energy, or the energy of motion, at higher temperatures. So if we compare the same molecule, illustrated in red here, at two very different temperatures, 4 degrees Celsius, which is about the temperature of the inside of your refrigerator, and then of course 37 degrees Celsius, which is your normal body temperature, you can appreciate that this same molecule is moving much faster at the higher temperature. So temperature's effect on membrane diffusion is not really going to matter a whole lot as long as the body is doing a good job of maintaining temperature homeostasis. The second factor we'll discuss is the diffusion distance. Practically speaking here, we're talking about how thick a cell is. So the idea here is a very simple one to grasp. The further a molecule needs to travel, the longer it's going to take to get there. Illustrated here, we have what we would consider to be a very thin cell. You could almost think of this cell as being flat like a pancake and then rotated 90 degrees as it's shown here. You can see that this cell must be very flat because the intracellular fluid space is very, very, very narrow. So this molecule, in crossing from one side of the cell to the other, is going to have to cross both the apical membrane and the basolateral membrane on the left and right respectively, and since this cell is so thin, it really doesn't take this molecule very long to get from one side to the other. But if we look at a different situation where the cell is now much thicker and the intracellular fluid space is much more vast, this molecule is going to take a lot more time to get from one side of the cell to the other. So this is going to come up when we start discussing different types of epithelial cells in a few videos, because the thickness of different types of epithelia is absolutely going to have an effect on the efficiency and speed of membrane transport processes in places like the lungs and the intestines. Next, we will talk about membrane surface area, which is a little bit of a different issue than cell thickness. So, higher surface area is going to speed up diffusion by providing more opportunity for diffusion to occur. You could almost think of two different cells that are roughly the same size, but have two different amounts of membrane covering their surface. On the left, you see an epithelial cell that has microvilli, which are microscopic projecting structures that emanate from the apical membrane, and structures like these serve the major purpose of increasing the surface area at the apical membrane and allowing membrane transport to occur much more quickly. On the right side, you can see a very similar cell that does not have these microvilli and therefore does not have as much membrane covering the surface of the apical side. And what we're going to see here is that we can't get nearly as many of these red solutes to cross the membrane of the cell that does not have microvilli, whereas the cell that does have microvilli can much more efficiently transport these molecules across the membrane, because since there is a greater surface area, there are more opportunities for these molecules to cross from one side to the other. So the idea here is that maximizing surface area for a given cell is going to maximize the efficiency by which these membrane transport processes can occur. And finally, possibly the most important of these factors that affect diffusion rate is what we call concentration gradient. A concentration gradient is just measured as the difference in concentration for a particular solute between the two sides of the membrane. This is not only going to influence the rate or speed of the diffusion process, but it's also going to dictate the net direction in which these molecules are going to move. If you look at this illustration here, you can clearly see that the red solute molecules here are much more highly concentrated on the left side of the membrane than the right. So we have a high concentration of the solute on the left and a low concentration on the right. The idea here is that the solutes are always going to show net diffusion from the area of high concentration to the area of low concentration. So these molecules are going to want to move in a net direction from the left side to the right side until we establish equilibrium. You can see this illustrated here. The net movement is from left to right, but there will be a handful of molecules that move from right to left. But once we establish equilibrium here, as you can see, in which now both sides of the membrane have an equal concentration of this solute, now the net diffusion is going to be zero, meaning the number of molecules that move from left to right will equal the number of molecules moving from right to left. So what we have just described with simple diffusion and talking about Fick's law of diffusion, this describes what is called passive transport. Basically, you can think of this as it being very natural for solutes to want to move from areas of high concentration to areas of low concentration. And because this is a natural process, you could almost think of it as being a downhill process. Passive transport does not require energy input. Simple diffusion and other types of passive transport that we will talk about do not require any energy input because it is just the natural inclination of these solutes to want to move from areas in which they're very highly concentrated to areas in which they're very lowly concentrated. So there are two types of passive transport, which again both involve molecules moving from high to low concentration. We, of course, have already discussed simple diffusion. This is a type of passive transport that applies specifically to solutes that can pass through the membrane freely. The other type of passive transport, which we will also talk about here shortly, is called facilitated diffusion. This, again, also applies to molecules moving from high to low concentration, but this specifically is talking about solutes that cannot get through the membrane all by themselves, and they are going to require some assistance to pass through, either because these solutes are too polar or because they're electrically charged. So for simple diffusion, again, this is going to apply to small, nonpolar solutes that move down their concentration gradient. In this illustration, you can see these blue solute particles are very highly concentrated on this side of the membrane, and over time, the net diffusion is going to pull them from this side to this side until we get to the point where equilibrium is established and the concentration of this solute is equal on both sides of the membrane. Once equilibrium is established, the rate of movement back and forth will have a net effect of zero. Let's look at a specific physiological example of simple diffusion. The illustration that you're looking at here is in the lungs. This structure that you see right here, this hollow-looking structure, is called an alveolus. Your lungs are made of thousands of individual alveoli, which are basically little inflatable sacs that when you take a big deep of breath in, these sacs of air inflate with oxygen-rich air. These alveolar sacs are bound on either side by a very special type of epithelial cell called type 1 alveolar cells. You will notice that these type 1 alveolar cells are very flat, and as we discussed earlier, very flat cells are really good at membrane transport processes. So the idea here is that when you take a big, deep breath in, this alveolus is going to inflate with inhaled air. The air that you inhale is very highly enriched in oxygen and has a relatively low concentration of carbon dioxide. This alveolar sac is very nearby a nearby capillary, and the capillary that is transporting blood through this area from the from the right side of the heart coming in through the pulmonary arteries, this blood is going to have a low concentration of oxygen and a relatively high concentration of carbon dioxide. This blood is coming from the body after the blood has already been depleted of oxygen from cells that have taken the oxygen away. So what we're trying to accomplish here in the lungs is we want to replenish the blood with fresh oxygen from the air that we breathe, and we want to rid ourselves of some of that excess carbon dioxide, and we're going to breathe that carbon dioxide out. So the question becomes, how are we going to do this? Well, we're going to accomplish this with simple diffusion. We're going to see that there are two very strong concentration gradients here, one for oxygen and one for carbon dioxide. The concentration gradient for oxygen points in this direction here from the inside of the alveolus across the membranes of the type 1 alveolar cells and the capillary endothelia and into the bloodstream. So oxygen will diffuse across the membranes of these epithelial and endothelial cells directly into the bloodstream where it will be bound by hemoglobin. The concentration for carbon dioxide is pointed in the opposite direction, from the blood plasma into the inside of the alveola. So the idea here is that because oxygen and carbon dioxide are both small, nonpolar, electrically neutral molecules, they can freely move through these membranes at a very high rate of diffusion, so this process is very efficient and occurs very quickly. So that by the time this blood moves away from the lungs and back to the left side of the heart through the pulmonary vein, this blood is now enriched in oxygen, high in oxygen, low in carbon dioxide, and that is going to be the blood that then gets pumped out through the aorta and distributed to all the tissues and all the cells in the body that need oxygen for cellular respiration. Speaking of which, let's take a look at simple diffusion gas exchange occurring in tissues and cells throughout the body that need that oxygen that's being delivered. So the idea here is that metabolically active cells throughout the body, such as skeletal muscle, for example, they are constantly consuming oxygen and emitting carbon dioxide through cellular respiration activities in their mitochondria. The consequence of this is that this is going to maintain in the surrounding interstitial fluid low concentrations of oxygen and high concentrations of carbon dioxide. So, as we said, the blood that is being pumped out from the aorta and into these systemic, highly active tissues is going to be high in oxygen and low in carbon dioxide. So here you can see the concentration gradients are flipped. There is a concentration gradient for oxygen that will cause oxygen to dissociate from hemoglobin, diffuse across the endothelial membranes of the capillary and into the interstitial fluid, where it can then be delivered to these metabolically active cells that need the oxygen to synthesize ATP through cellular respiration. As the cells perform cellular respiration, of course, they produce a lot of carbon dioxide, which is removed from the area because there is a separate concentration gradient for carbon dioxide from the interstitial fluid into the bloodstream. And now, this excess amount of carbon dioxide can then be carried away back to the lungs, where it will then do exactly what we saw on the previous slide. So again, the reason why oxygen and carbon dioxide can do this type of membrane transport called simple diffusion is because they are small, they are nonpolar. They conform to every single kind of rule that we established in our previous video. So the other thing I want you to notice here is that the concentration gradients for both oxygen and carbon dioxide are opposite of one another. They tend to move in opposite directions. So that's another important lesson. Just because oxygen is moving in one direction, that does not mean that carbon dioxide has to move in the same direction. The movement of any given molecule is dictated by the concentration gradient just for that molecule. The concentration gradient for all the other molecules is pretty much irrelevant. Okay, so let's move on to talk about the other type of passive transport called facilitated diffusion. In the case of facilitated diffusion, we are of course considering molecules and solutes that are not quite as lucky as oxygen and carbon dioxide. They can't freely pass through the membrane for whatever reason, either usually because they are too polar in the case of something like glucose, or because they're electrically charged in the case of things like sodium and potassium and calcium. So, as we said in the previous video, that does not mean that those sorts of solutes will never ever pass through the membrane. It just means that they're going to need some assistance. But the question is, what is the actual molecular form that this assistance takes? So, the answer is channels and carriers, two different types of integral membrane proteins. So channels are partially selective based on the size and charge of the solute, and as such, channel proteins are usually always going to be transporting electrically charged ions. We saw an example of this in a previous video. This ion channel produces a water-filled channel through which the ions can move and avoid having to interact with the hydrophobic tails of the phospholipid bilayer. In the case of the channels as well, it's important to consider that channels are not necessarily always open. Some of them are, but some of them will open and close based on specific physiological stimuli. In fact, we will see in the future, a lot of channels are linked up to receptor proteins, and these channels will only open when that receptor is bound by a very specific type of either a neurotransmitter or a hormone. So something to keep in mind for now. Carriers are going to be a lot more strict about the type of solute they transport. They're a little bit similar to enzymes in that a particular type of solute they're going to transport conforms to a shape of something that looks a little bit like an active site. This carrier that's illustrated here, you can see is very particular about binding to something that is shaped like glucose. So glucose in this case binds to the carrier, and then the carrier goes through a type of shape change that allows glucose to be released on the other side of the membrane. Now, this is probably going to seem a little bit different compared to the simple diffusion that we saw before, where molecules move freely through the membrane, but really there's not that much different here. In both cases, you have a solute moving from an area of high concentration to an area of low concentration, both for the channel and for the carrier. The only difference here is that these molecules, when they move from high to low concentration, they just need something to facilitate their passage through, something to give them a little bit of help, whether it's the channel or the carrier. So the idea is that even though assistance is being provided here, there is still no energy input that is necessary because the solutes are still moving downhill from high to low concentration down their concentration gradients. Okay, that's going to do it for this video. Here is a list of vocabulary terms you probably ought to be aware of after watching this video. For checking your understanding, number 1, what dictates the net direction a molecule diffuses across the membrane? Number 2, do all solutes follow the same concentration gradient, or do they move according to their own gradients? And 3, for solutes that cannot cross the membrane by simple diffusion, how do they get across and what is necessary? Alright, that's going to do it for this video. If you have any questions, go ahead and drop them in the comments section, and I will see you next time. So long.